Professor Technologist Dr. Nazip Suratman, UiTM Director of Research Nexus, Professor Dr. Noritawati Mat Tahir, our invited speaker, Dr. Dina Jane Kling, President of the Fulbright Alumni Association Malaysia, Dr. Fakhrul, Hazman, Yusoff, lecturers, researchers, undergraduate, postgraduate, distinguished guests, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to all, and night for those in US. Okay.
It's an evening
Alright.
in US, yeah.
Okay, first of all, let me introduce myself. I'm Abdul Hadi Abdul Razak, the coordinator of Cyber Technology, Research Nexus UiTM, and I'll be your moderator for today's online seminar. Okay, on behalf of TNCPI Office and Fulbright Alumni Association Malaysia, I would like to welcome everyone here to Fulbright Webinar Sharing Session. Okay, I'm pretty sure that this webinar could serve as a platform for everyone to explore into a multidisciplinary research and academic nexus. Okay, so without further delay, I would like to warmly invite Dr. Fakhrul Hazman Yusoff, the President of Fulbright Alumni Association FAAM, Malaysia FAAM, for his welcoming speech. Please welcome Dr. Fakhrul.
Okay, thank you, Dr. Hadi, my friend Dr. Hadi. So, Assalamualaikum warahmatullahi, and a very good morning, everyone, and also good evening for those who are attending from US. First of all, on behalf of Fulbright Alumni Association Malaysia and Faculty of Computer and Mathematical Sciences UiTM, because I'm also a member of the faculty, I would like to thank Prof. Dr. Nazip, the Deputy Vice-Chancellor on Research and Innovation, UiTM, and also UiTM Global and Center for Computer Science Study, Faculty of Computer and Mathematical Sciences, UiTM, for their support in ensuring the success of this event. I also would like to welcome our speaker, Dr. Dina J. Kling, to this sharing session, or should I say welcome home? Because I think Malaysia has become your adopted country now, given that you have done project here in Malaysia, and I believe you will be having more projects here. So welcome and welcome home, or something like that. And speaking about this event, the event is aimed for the researchers to get an overview of how research can be conducted and analyzed using machine learning as the analytical tool. So you use the machine learning, for your research. We try not to overcomplicate the ML with too much theory, as ML tools nowadays are relatively simple. It's not like the old days. The old days you have to install and set up on your own. You have to know all the bare bones. But nowadays there are tools to do that. But we need to know what you're using, right? If you click here, you click there, you should know what you're clicking. It's not just like you ended at the scamming place or something like that. So it's a relatively simple, at least for a certain aspect of the research, and definitely those who are in computer science, those who want to go deeper, they should learn more on the elaboration of the theory. Welcome all the audience. I hope that this event can be beneficial to the audience in terms of applying machine learning in whatever field they are doing. Right. That's all from me. Thank you, and back to you, Hadi.
Hello. Okay.
Yeah.
Thank you very much, Dr. Fakhrul, for a refreshing morning welcoming speech. Okay, next on the agenda, I think I would like to heartily invite UiTM Deputy Vice-Chancellor of Research and Innovation, Professor Technologist Dr. Nazip Suratman, for his welcoming remarks. Ladies and gentlemen, please join me in welcoming Prof. Nazip. Prof. Nazip.
Bismillahirrahmanirrahim. Thank you very much, Dr. Abdul Hadi. Very good morning, and can you hear me clearly, everyone?
Yes, we do. Yes.
Yeah. Very good morning and good evening, everyone. Distinguished guests, ladies and gentlemen. First and foremost, I would like to greet all attendees and guests to the Fulbright webinar sharing session this morning. The Office of Deputy Vice-Chancellor, Research and Innovations is hosting this webinar in collaboration with the Fulbright Alumni Association Malaysia, FAAM, UiTM Global, and also the UiTM's the Faculty of Computer and Mathematical Science. Welcome everyone. I envision this virtual session as a forum for academicians, for researchers, for the graduate students and university professionals to network and make contacts with one another. This is also an excellent time for us to exchange ideas with subject matter from the experts and from the researchers related to the subject. Today, we are deeply honored. We will hear from Dr. Dina Jane Klink of the K. Lisa Young Center for Conservation's Bioacoustics Cornell Labs of Ornithology, USA, which many of researchers and postgraduates from various backgrounds, preliminary, engineering, computer science, forestry, social science would be drawn to her presentations. Ladies and gentlemen and distinguished guests, let me share you briefly on the strategic goal of UiTM. The goal of UiTM, as stated in our UiTM Strategic Plan 2035, is to empower research and innovations to become a globally world-renowned university by the year 2025. This goal necessitates a concerted efforts which include raising academic standard to a global level. Furthermore, stemming from the research that can accelerate development is quite challenging, but it will eventually benefit society and the economy. Ladies and gentlemen, I sincerely hope that this session will provide more opportunities for UiTM researchers to engage in international collaborations, particularly with experts in their respective fields. And I'm confident that an event like this can help participants benefit from one another. I would like to encourage researchers to apply for the Fulbright grant, scholarships, or any other programs that are available. This will undoubtedly provide you with international exposure, which can serve as a foundation for your future research to fly at a higher altitude. Ladies and gentlemen, before I conclude, I would like to extend an official invitation to Dr. Klink to visit our University Tuan Haji Mara research stations in Pahang National Park in Kuala Kenyam. This is an ideal setting for multidisciplinary research focusing on biodiversity, on the community, and environmental sciences. So we have numerous opportunities to collaborate in this area, and I would like to share that currently we have collaborated with our US counterparts from the University of Harvard, University of California, Santa Barbara, from Dartmouth College, who are working on the indigenous community in National Park. Other than that, we also have collaborators from Austria, Academy of Science Austria. We have someone from University of Kastamonu, Turkey to study on plants. Okay? So again, I'd like to extend warm welcome to everyone, to Dr. Fakhrul as well, to enjoy the complex ecosystem of tropical rainforest in Taman Negara National Park. On that note, I would like to thank again the organizing committee, especially Dr. Fakhrul, the presidents of the Fulbright Alumni Association Malaysia. Well done, Dr. Fakhrul, keep it up, for initiating contact and facilitating the realizations of this webinar. The time and effort put into this thoughtful discussion will benefit all parties involved. Thank you very much. and have a fruitful and beneficial webinar. Thank you.
Okay, thank you, Prof. Nazip, for a precise and meaningful remarks for all the audience here. Okay, I would also like to take this opportunity to thank the Office of Deputy Vice-Chancellor, Research and Innovation, UiTM Research Nexus, and of course FAAM for making this virtual event possible. Ladies and gentlemen, today we are fortunate to have Dr. Dina Jane Kling from Cornell University as our guest speaker in sharing her personal research experiences. Okay, before I invite our speaker, allow me to share Dr. Kling's brief background. Okay, Dr. Kling currently is a postdoctoral scientist at Cornell University on a collaborative project between the Nature Conservancy and Cornell Atkinson Center for a Sustainable Future. She obtained her PhD in evolutionary anthropology with specialization in primatology from University of California, Davis in 2017. Dr. Kling recently completed a Fulbright Asian US Scholar Grant where she held a joint position as a lab associate with Bioacoustic Research Program at Cornell University. Her principal scientific interest is in the behavior, ecology, and evolution of acoustic signal. Her research focuses on primates acoustic communication from comparative and evolutionary perspective. Her aim to answer related to evolution and maintenance of vocal diversity in primates using innovative bioacoustic technique with emphasis on testing new technology and drawing from diverse fields such as human speech recognition, machine learning, and signal processing. She particularly interested in evolutionary mechanisms that shape variation in primate vocal communication system and understanding the function of primate vocalization, as this can help us understand the evolution of communication in our own species. A very comprehensive background for someone quite young. Okay, ladies and gentlemen, before I hand this mic to Dr. Dina, you are welcome to write down your queries relevant to this topic in the chat box, and I'll address Dr. Kling at the end of the session. Okay, so without further ado, I would like to invite Dr. Dina Jane Kling to give her speech, which entitled Machine Learning Approach Provide Insight into Behavior, Ecology, and Conservation of Non-Human Primates. Dr. Kling, the floor is all yours.
All right, thank you. Can you hear me all right?
Yes, clearly.
Yes. Okay, wonderful. Oh, well, thank you so much for that. Just wonderful introduction. And yeah, really, I am missing Malaysia as my home a lot right now because obviously it's been quite a while since I've been able to travel there. But, you know, it's so wonderful that we have these virtual things that we can do to continue to work together and make new connections. And so this is the first time I've used WebEx. So I will try to do the screen share, but you'll have to give me some feedback if it's working. Okay.
Sure.
Okay, is it working? Oh, let's see. It says that I didn't give the right permission. Sorry. Has anybody done the permissions before? It's telling me that I didn't give it the permission to share.
Are you using Mac or Windows?
A Mac, yeah.
I think you need to go into the system preferences. You have to allow this Cisco WebEx to share your content.
Okay, let's see.
But I can't remember how did I do it.
And so is that under security?
Yes, I think so. I can't remember. Wait, let me check.
Let's see. Normally it'll pop up and nothing popped up. Okay, let's see if I should say it again. Okay, so then the host there. No, sorry, still having technical difficulties.
Usually, if you first time use it, when you share that, they will automatically open the
Yeah.
screen. Yeah, that's the thing. Maybe others can help.
Let's see. Let me look and see. I'm just using the web browser. I don't know if that will be a problem or not.
If it's possible, I think if you can send the slide, maybe I can share it.
Okay, yeah. Let's see.
I type my email address at the chat.
Yeah, sorry about that. We use Zoom all the time.
I think. Can you try now? Because someone tell me that you're already allowed to share. Try to share your slide again. Before, if not, we try other option.
Says it's connecting. It's doing something different now, so maybe it's gonna work. It's just doing the little like bar thinking thing now.
Is it possible?
It's a really big file, and so it's just giving it's just sharing content. It says share content and connecting, and it's not doing anything. So that's really odd. Yeah, I could try to log out and download the app really quick.
I think take time. I think if better option if you send it, email me the slide, then I'll share it. Is it better that way? Is it okay?
Yeah, I'm fine with that, but it's a very big file too.
Oh, is it?
Videos and sounds in it, so.
Okay.
Yeah, it's just saying share content and it's not doing anything now. So I could try to refresh to see if it will do something. That might kick me out. Let's see.
Other option, maybe you can share a Google link drive or something, just to, just for.
I know, yeah, just trying to get it going. Let's see. Yeah, it's uploading, but it's taking quite a while.
It's okay. Take your time.
Yeah, sorry. I can try to download the app, maybe the WebEx. Mac. Or I can also try to log out and log back in and see if that helps. Yeah, it's just when you say share. Okay.
Sorry, audience, we have some technical problem, but I think we can solve it in the meantime.
So I just downloaded the app, and it says to put in a meeting link, and so I just put in. Oh, I wonder if it's just this one.
If you already install the apps, I think you just click the link, they automatically open it.
Okay, all right. Let me try one more thing. So.
All right.
Okay, it's okay.
I think
Okay.
it's finally. I'm so sorry.
Magically, it's open.
I thought it was never gonna happen. I'm so sorry.
It's okay. It's okay.
All right. Okay.
Okay.
So,
All right.
Okay. So whenever you're ready, you can begin your
Okay.
presentation.
Yes. Thank
Okay.
you.
All right.
Thank you so much for your patience. That's one of the longest technical difficulties that I've had so far. But let's hope that that we don't have any more between now and the end of this session. So, anyways, as I was saying, really thank you so much for inviting me here, and it's really just such an honor. And yeah, I have spent almost 2 years of my life in Malaysia, mostly on Malaysian Borneo and Sabah. But I'm hoping to start to spend more time in Peninsular Malaysia. And so I've been working with mostly University of Malaysia Sabah, but then now hopefully soon I can add another logo here with some other universities and maybe UiTM. That would be wonderful. So, and I do also want to just mention the Fulbright and how it has been just pivotal in kind of my success as a scientist, and also, you know, in the basically my kind of growth as a human being as well, because they really do emphasize kind of cultural exchange. And the Fulbright is such a fantastic program. And so if anybody is interested in applying for the Fulbright to come to the US, and you have questions about that, I'd be happy to talk more about coming to the US and trying to help you pick where you would go, etc. So please just know that I'm here. And so now I'll delve into the talk that I'm going to be giving today. And so in the introduction that you heard, my background is really in primatology or evolutionary anthropology. And it was kind of an interesting, funny road that I took to end up getting into more of the data analysis and machine learning parts. And so you may be thinking, like, how does behavioral data collection of gibbons kind of fit with applying machine learning techniques? And I hope that in the talk that I give today that will kind of show you kind of how this all comes together, and how even if we're studying non-human primates, that really these days we have to really rely on these more kind of sophisticated data analysis techniques. And there really is, particularly in this field, such a wonderful kind of area or place for synergies and interdisciplinary work. And so now, so I always like to start my talks by either playing some sounds or here I have a video. And so a lot of you are probably familiar with what I'm going to play and show you today. Oftentimes the audiences that I have in the US are not. So I will go ahead and I'm going to play the video. And I want to give one little caveat here, is that I tend to work with animals that are not habituated. So that means they're not used to humans. And that means I don't generally get very close to them. And so this video is maybe not like what you would see on like BBC or National Geographic. But that's also because like I was just there right underneath the tree, and this is about as close as we get to animals that aren't used to people. So I'll go ahead and play this real quick. And I'm not sure, I can't see the chat, so I was going to maybe ask if people could identify what that is, but maybe just in your mind, if you know, you could tell yourself. And so this is, they actually were recently just transitioned to their own species. It's the northern gray gibbon. They're Hylobates funereus, and they're found in Sabah. And so this was a male gibbon, and so males will sing solos early in the morning about sunrise, and then the males and female gibbons will also sing together. They'll do what we call a duet, where they will alternate back and forth in this really complex interaction. And so when I first went to Sabah for my dissertation work, it was in the summer of 2012. And I had all of these plans to do full day behavioral follows. And so that's what primatologists, tend to do if they want to get behavioral data. And so this requires you get up way before sunrise, because you have to get to the sleeping tree of the primates, and then you follow them all day from sunrise to sunset, and you take down all of the behaviors that they're doing. And so my plans were to start to try to take some behavioral data from gibbons. And so I'm not sure how many of you have gone into the forests of Sabah before. They are very hard to navigate often, and a lot of times there are lots of cliffs and they're really steep. And so I went out and I was like, okay, I'm going to go follow these gibbons and start to collect behavioral data. My first day out there, I go and I saw them actually, and I was like, all right, I'm going to follow them. I try to follow them and they take off because they're not used to humans and they were nervous. And so they take off and I'm like, all right, I'm going to follow them. And then I just came to a big cliff and I couldn't collect the data. And so I was like, okay, what am I going to do? Like, how am I going to, I'm out here, I want to collect some sort of data. What can I do? And so I came back to the field center and I was looking on my computer because I had all of these papers saved. And I was like, okay, so what kind of data can I collect? And I was reading papers that people had written about studying gibbon vocalizations. And so they are really well known for these loud calls that you've heard, and maybe you've actually heard in the wild as well. And so I always like to start my talks kind of telling people, science, a lot of it is just kind of luck and serendipity. You can go in with all of these grandiose plans, but a lot of times, especially if you're working with animals, they may or may not follow along with those plans. And so you need to be flexible, but then in the end, you never actually know how it might turn out. And so in my case, I got really, really lucky that I was working with a really vocal species. And now I study bioacoustics, which is basically the study of animal sounds and the habitats where they live. And so the title of my talk has the term machine learning in it. And you may have heard this term before. And so I wanted to just take a moment and define it as we're going forward so that we all have kind of an idea of what I mean when I say machine learning. And so I'm going to include, there's a couple, when I was doing a search in preparation for this talk, there are actually many different definitions of machine learning. And so I'm including just a couple here. And so one is machine learning is the science of getting computers to learn and act like humans do. And so that was one that to me felt a little bit kind of human-centric and not exactly kind of fit for what I think of machine learning as being. So the next one, machine learning is the science of getting computers to act without being explicitly programmed. Okay, but that's still not a very precise definition. And so then the last one, and the one that I found that fit best with my understanding and use of machine learning is that machine learning is the study of computer algorithms that improve automatically through experience and by the use of data. And it's actually part of the broader field of artificial intelligence. And so this is where I think people get a little bit confused because sometimes people will use these terms interchangeably, but machine learning actually falls within the broader field of artificial intelligence. And so here we have just our working definition, which I can go ahead and move on to the next slide. And so machine learning, regardless of the systems that you're working in, and so machine learning can be used on all sorts of different types of data. And so image classification is one, sound classification, that's another one. And so, of course, because I study animal sounds, we focus more on the sound classification part. But when you're using machine learning, there are generally two different types of approaches that we use, and I'll talk you through the definitions of these, and then I'll give you specific examples as we move forward through the talk. So the first one, supervised learning or supervised classification, is that with supervised learning or supervised classification, in this case, we have labeled or ground truth training data. And so the example that I'm going to use for this talk today is that I've been really interested in trying to look at differences between individual gibbons based on their songs. And so in this case, the supervised data set will have, for each call, I will know which individual animal was singing, and that will be a label that I input into the algorithm. The alternative to this is unsupervised learning. And so this is basically an approach where if we use it if we do not have labeled training data. And so in this case, the goal is to try to understand or infer natural structure within the data set. And so the example that I'm going to be talking you through today is a similar one where we're interested in individual variation in animal calls. But in this case, the data are collected in a way where we don't have the human labels or the labels for who's calling. And so we input the data and we ask the computer to tell us how many individuals there are in our data set. And so I'll revisit these two ideas, but supervised is that we have labeled training data. Unsupervised means that when we input the data into the algorithm, it does not require labels for the training data set. And I know that this is a really diverse group, and I know that you probably have kind of different levels of experience with bioacoustics. Maybe some of you have never even heard of it before. So I want to make sure that we're all kind of on the same page here. And so bioacoustics is basically the study of animal sounds and the sounds of the habitats where the animals live. It's a really, as I mentioned, interdisciplinary field. So we have the behavioral ecologists, so the people like me. We have engineers, the ones that are putting together the recording devices and the ones that are developing the software that we use to analyze the data. So we have all of these different people coming together, and actually within our lab as well, we have a really diverse group of people with a lot of different areas of strength and expertise. And so it's really fun to give a talk like this to people who are actually from Southeast Asia, because you have a pretty good idea of the lay of the land and the geography here. But just to give you an orientation about where the majority of my data up until now have been collected from is in Sabah, so in the northern part of Borneo there. And so as somebody that studies animal sounds, you would think that I would listen to the sounds a lot, and that's actually not the case, which may come as a surprise to you. And so what we do is we generally, if we're looking for signals of interest, we generally create what are known as spectrograms. And so I will be showing you lots of different spectrograms as we are going through the rest of this talk here. And so I want to just orient you to what I mean when I say a spectrogram. And so we can think of a spectrogram as it's a visual representation of the sound. And so here we have two different spectrograms that come from two different gibbon female individuals. And so on the y-axis, we have time. On the x-axis, it's the frequency or the pitch of the sound. And then the color represents the amplitude or the strength of the signal at a particular time and frequency. And so hopefully I can go ahead and play this for you now. Could I maybe have somebody just give me a thumbs up or let me know if they can hear this? Okay, thank you. Thumbs up. Okay, so that was one gibbon female individual. And so I want you to note that the structure of the call, it tends to go from these longer notes to these shorter notes. But there also is variation kind of in the number of long notes versus short notes. And then if you look at what we call the bandwidth, so the frequency range of the notes actually changes too. And so now I would like to have you listen to the second one and see if you can, or listen and hear if there are differences, noticeable differences. Some of my favorite sounds. I've spent a lot of time doing listening to these and looking at spectrograms of these. And so as I mentioned, the gibbons, they duet. So the males and females will go back and forth, but the female has this really distinct, easy to hear, easy to pick out call. And so that's a lot of my work has focused on that, mostly just because it is so stereotyped. So it's one of the easiest parts of the calls to pick out. And so I'm sure at some point you have heard about sound and what sound is, but I want to just give you a little bit of background on sound as a form of digital data. And so when we go out with, if I go out into the forest, I have either a handheld battery operated recorder, or now we're switching to these autonomous recorders that can go for 6 months at a time, where you just kind of plop them out into the forest. But you can think of sound as a wave that is traveling through the environment, and it's going through some medium. So water is one, and sound can travel actually really, really far underwater. And then, of course, air. And so what happens is that when the sound reaches the microphone, it's actually coming in as a wave. And the microphone is picking up the vibrations at a certain rate, and that's something that you can set. And so it's basically taking data every so often on how the sound pressure is changing. And so the most common sample rate that we use is 44.1 kilohertz. And so what that means is that per second, the recorder is taking data 44,100 times. And so if you think about, this is a 8-second long call here. If we are taking 44,000 samples every second, and then you times that by 8, that's going to be a lot of numbers. And so you don't need to worry about the specifics of that. But the point that I want you to know is that I'll go ahead. So now this is what we call a waveform. And so this is the digital representation of the recording that I just showed you. And so if we zoom in, so the time axis on the first one was 8 seconds. And now we're zooming in, and this is about 0.1 seconds, and you can start to see the wave-like structure of the sound wave. And then now if we zoom in, so this is a really short time fragment here, and then zoom in a little bit more. And so this is a plot that I just made in R. So I use R for almost all of my data analysis. And And so you can just zoom in and you can see, oh, actually, like any recording that you take, any digital audio recording, it's basically just taking a bunch of numbers. And so as researchers and scientists and computer scientists in particular, we need to, once we have these data, we have to figure out, okay, what are we going to do with all of these data? Because if you think about just for an 8-second sample, if you're going to have, hundreds of thousands of values, we have to think about a way that we can condense those values down into something that's meaningful and useful. And so the most common way that we do that, and I showed you this already, is the spectrogram. And so when we create the spectrogram, this does actually reduce the data a little bit in terms of the overall values that we have for each call. And so I'll go ahead and I'm going to zoom in on these just like I did on the other ones, just to give you an idea of what I mean when I say it's kind of reducing the data. So again, the spectrograms, we have time on the x-axis and frequency on the y. And then it's actually for each time and frequency bin, it's calculating a number using the fast Fourier transform, which you don't need to worry about the math that's happening there underneath, but just know that that's what it's doing. And so if we zoom in, so now we're zooming in onto the time even more. So we went from 8 seconds to 2 seconds. And now this is about 1 second. And so I didn't put the numbers in here because it was too messy for you to get the point. But what's happening is that each box here is actually, the color is representing a number or a value. And so when we create a spectrogram, we go from the original waveform and we can reduce the values that are representing the signal into, it's about half, depending on the settings that you use for the spectrogram. But if you think about, so for one call, so we have one gibbon call here. If we go from, like over 500,000 samples down to maybe like 50,000 samples per call, if anybody's tried to do any sort of modeling or machine learning or anything with that, if you know that you have multiple calls in your data set, and for each call you have 50,000 values that are representing that call, that's still going to be way too much for the computer to handle, basically. And so that's why, as scientists, we need to think about kind of how we can take these big data sets and condense them down into useful and meaningful information. And so I wanted to just include this plot here down at the bottom. And so this is from the Stability of Altered Forest Ecosystems project in Sabah. And I set some recorders out to just go for it was my first time doing pilots work, and I was out there for about 2 weeks. And so this is showing here, as opposed to on the duration of, on the time frame of about 8 seconds. This is showing kind of what's happening in the soundscape, or the overall kind of acoustic area over the course of 12 days. And so again, if we think about, if we have multiple recorders out that are recording for 24 hours a day, that are recording for 6 months or more at a time, that comes into a really big data question. And, what do we do? How do we get useful and meaningful information out of that data? And so that's a really big thing that we are working on in our lab. And I'll talk you through a couple different approaches that we're taking. And it really also depends on the question that you're asking. But so one of the major considerations when we're thinking about analyzing acoustic data is, which features of the acoustic data set should I use in order for the analyses that I need to do? And then the next one, so coming back to the idea of machine learning, we can think about, so what are my research questions? So what kinds of questions am I asking? And then would supervised or unsupervised classification approaches make more sense? And so if you remember, supervised classification approaches, those have labeled training data, whereas unsupervised classification approaches do not have labeled training data. And so I mentioned this a little bit already, and something that I have done a lot of work on is the use of supervised learning, and using that to see how well we can distinguish or tell between different individuals of the same species. And so a lot of the early work that I was doing something that we call feature extraction from the spectrogram. And so this is from one of my first paper of my dissertation that came out in 2017. And it's showing on the spectrogram here. So I've showed you a couple different spectrogram kind of versions of the female gibbon call, and they're just made in different programs, but this is from the publication to show that how we extracted features from the spectrogram. And so this is something that I did it's a program called Raven that people or engineers in our lab actually developed that help you to create the spectrograms, and then you go in and you manually draw boxes around the calls of interest, and then you can pull out all of the relevant information. So we were interested in looking at the duration of the total call, the duration of the part with the shorter notes, the duration of the part with the longer notes, and then the subset part up at the top there, it's showing that's the longer notes. And so for each of those, we looked at the maximum frequency, so how loud that note got, and then the bandwidth, which means basically the frequency range. And so my justification for starting to get into this was thinking about how we can use individual vocal signatures for conservation, and I'll talk about that in a few slides. And in doing this, I got some really great information, but it was not a quick or straightforward thing to do. For the data set in this paper, we had about over 300 calls from different gibbon females, and for each call, it took me about 10 minutes to do what we call annotating or extract features from the spectrogram. And so this is something that's okay for academics to do as you're writing your dissertation, but not something that people that are out in the real world that want to know how many individuals there are in a particular area, they're not going to want to do that just because it's not practical. But I'll talk about in the next slide some different ways that we've been trying to do that. But I did get some really cool results from this paper. And so coming back to the idea of supervised classification. And so in this instance, I was there with a handheld microphone underneath the gibbons while they were calling. So I was able to see who was calling. I took the GPS point. I saw how many animals were in the group. And then gibbons are territorial. So that means if you move quite a far distance away, we can assume that it's going to be a different group because they tend to stay within their own home ranges and they don't really move. And so in that case, I had data that was okay to use for supervised classification because I had the labels that said this is from female A and female B and female C. And so I was really happy. So I used a supervised classification algorithm called linear discriminant function analysis. And so with this one, and I did what we call leave one out cross-validation. And so I would run the LDA, or the linear discriminant function analysis. I would run it on all of the calls except for one. And then I would plug that call back in and I would have it predict which female that call came from. And by doing that, by validating using this leave one out cross-validation, I found that we were able to classify with over 90% accuracy with 30 plus females. So a really, really high classification accuracy, which we were really happy about. And so basically what this means is that the gibbon female great calls are individually distinct. So we do have these individual vocal signatures. And so just to talk you through this plot really quick. So what we're seeing here, it's called a biplot. And so when we run the linear discriminant function analysis, it outputs what we call the linear discriminant functions. And so this is the first and second one that we're plotting here. And so on the top plot, each point represents one call from one female. And then the colors represent the different females that were in our data set. And the circles around are the 90% confidence intervals for a particular female. And so how we can interpret this is that because we're seeing separation of the calls out away from each other, we can basically infer that there is vocal individuality and that females are distinct from each other. the alternative, if there's not a lot of variation or individual variation, we would see that the points would be just on top of each other. My advisor always used to say it would look like a sneeze, like somebody just sneezed and all the points were on top of each other. And so we're not really seeing that. And then we found too that so the features that were most useful for distinguishing between individuals were not the frequency characteristics, so like the maximum frequency or the bandwidth, but it was the temporal features, so the duration of the notes and the duration of the different parts of the call. And so as I mentioned, the choice of features that we use matters. And so I just showed you a technique where we use feature extraction from the spectrogram. But then we also have a lot of other types of features that are available to us. And so for a paper that we put out in 2018, I tested another kind of feature, which I won't go into the details about what these features are doing, except for I just want you to know that they have been really, really useful for kind of like early human speech recognition applications. And then these features, you can calculate them really, really quickly in R. And so I mentioned that for my dissertation chapter where I was doing the feature extraction from the spectrogram, that it took me months of doing the data analysis and like looking at the spectrograms and drawing boxes around the notes. Whereas with these MFCCs, I have the code, I can read in the sound files, do it in R, and it'll process it in, like, depending on how many sound files you have, like 5 minutes or something. So I reduced the workload from months down to minutes, not to mention all of the time that it took me to actually get the code going. But any of you that are into coding, just know that, you know, those startup costs are worth it once you actually get the code running. And so, as I mentioned, this substantially reduced processing time. And then the other thing I was really happy about is that it increased our classification accuracy. So this was from the same data set that we had from the previous talk that I showed, or the previous paper I showed you, but it increased our classification accuracy a lot. So we were really, really happy about that. Okay, and so I kind of touched on this already, but I want to just kind of revisit this idea of how and why we would study gibbon individuality and how that could translate into conservation or conservation applications. And so you may or may not know that gibbons across Southeast Asia are endangered. And so gibbons are found not only in Malaysia, but also India, all the way to China, and then down south into Indonesia as well. But across the range where they're found, they're endangered. And for most conservation applications, having a clear estimate of the number of individuals in an area is really, really crucial for conservation. So, for example, if we need to know kind of how fires or deforestation for oil palm plantations or any of these kind of potential conservation impacts, if we want to know how they're impacting the animals, we need to know how many animals there are in an area. And generally we need to, in order to do that, we need to be able to ID individual animals. With some non-human primates, so I worked on a project in Argentina where we darted and radio collared owl monkeys. And so for some primates, you can dart and radio collar them. And so in that case, you can track them that way. But if anybody here has ever seen a gibbon in the wild, then you know that they are really, really agile and they're really, really high up in the trees as well. And so it's just really, it would be impossible to dart them ethically. And so that's another really big justification as to why we are really interested in working to develop this method to use individual vocal signatures to track and monitor gibbons non-invasively. Okay, and so now still staying within the realm of supervised learning, but I want to now kind of shift gears a little bit to another project that I've been working on developing, and then really people kind of across the field of bioacoustics have done all sorts of work to develop different types of automated detectors. And So the automated detector that I have developed is in the R programming environment, and it's a really, really basic one. So it kind of builds on what the early human speech recognition automated detectors were doing. And so now, if you have the computing power and if you have the training data, you can use the deep neural networks to create more effective or potentially better models for automated detectors and classifiers. But the really crucial point with that is that you do need to have a lot of training data. And so the more simplistic one that I'm going to show you here that relies on the machine learning type of stuff, this one, it requires some training data, but not on the kind of orders of magnitude that some of those other ones do. And so I'll talk you through just really briefly the steps that my detector takes, and then we'll go through and we'll play a little bit of a game where I will ask people to see if they can do better than my automated detector at identifying if it's a gibbon female or not. And so first, for my automated detector, the first step that has to happen is that, so we have, and as I've mentioned before, we have these battery operated or these autonomous recording devices that will go for hours and months at a time. And so the first thing that this detector does is that it goes through and it pulls out any potential sound events within our frequency range of interest. And so in my case, I'm interested in pulling out the gibbon calls. So I will focus only on potential sound events within the frequency range of the gibbon call. And then once we pull out any sort of signal, and so this is not kind of discriminating if it's going to be a gibbon or not at this point, it's basically anything that's making noise within the frequency range of the gibbon. The next thing we have to do is we need to take that waveform, so all of the numbers that are in the waveform, and we need to condense those numbers down into a smaller, more useful set that we can then apply and feed into our machine learning algorithms. And so we use the feature extraction for each sound event. And so in this case, I'm still using those MFCCs, the ones that I mentioned before, because they're a lot easier to use than the features from the spectrogram, and they work really, really well for gibbons. And so then the next step is we input these features for each of the sound events into our machine learning algorithms. And so I have been playing around with Gaussian mixture models and then also support vector machines. Those are the two ones that I am really familiar with that tend to do really, really well. And so at this point, we put in the sound event and we say, okay, so, is this a gibbon? Is it a hornbill, or is it an elephant, or, background noise or something else? And then the last step, and we're getting close with some automated detection type approaches where we don't need a lot of human validation of the system, but we're not at the point where we can reliably say that every detection that we have gotten from the detector is the signal of interest. And so we do need people to go back and at least spot check some of the detections to make sure that, okay, this says it's a gibbon, it is actually a gibbon, okay. Or, it says it's a gibbon, but it's an elephant or something else. So we are still at that step where we need human observers to help validate. And so if any of you have used any sort of voice recognition software before, so I have an iPhone, and so anytime I'll say, hey Siri, and it'll turn on, oops, and it just turned on just then. She doesn't understand. Okay. But you'll know that if you say something, a lot of times it's not picking up. So it's not 100% accurate. And so that's why when I've been playing around with my detectors, it's been actually quite fun to see what it picks out. So the question is, if you can do better than an automated detector. And so I'm not sure if I can have the chat, if I can see the chat while I'm sharing my screen. No, I don't know if I can. So I guess sometimes I like to try to have it be interactive, but maybe if somebody who can watch the chat and let me know if people are guessing, and you can just verbally tell me. So, oops. Let's try to, okay. So let me know if you think this is a female gibbon or not. And if you think it isn't, let me know what you think it might be. Okay, so here's a spectrogram. You've seen quite a few of them, and I'll go ahead and play this. I saw one guess for, so I'm seeing the chat pop up, so that's good. So I saw a guess for a female gibbon. I saw, okay, yes, I saw one. Oops. Helmeted hornbill. All right, how about the next one? Any ideas? Female gibbon, not female gibbon? I see one guess for a female gibbon. That one was a rhinoceros hornbill. All right, next one. Do I have any guesses? Female gibbon, not female gibbon? So this is actually, oops, a great Argus pheasant. And so they're found throughout Southeast Asia as well. So, and they're well known for, they clear these dancing grounds in the forest. And so they're actually more well known for their visual displays where the males will clear these dancing grounds. So they'll clear all the leaves and everything out. And I've walked through them before. It's like almost like it's very pristine in the forest. But then the males will like come up and run around the female and do all of this really like interesting stuff with their wings and everything. And so they're more well known for that, but they also are quite vocal. And my detectors love to pick up the great Argus pheasants because they're in the same frequency range and they have like similar kind of structure of notes to the female gibbon. So I get these as detections quite often. And it actually, I got them so much that I decided to write a paper about great Argus pheasant vocalization. So myself and one of my students that I was working with, we got a paper out that was looking at great Argus pheasant vocal behavior in Danum Valley Conservation Area. Okay, how about this one? Is that a gibbon? Not a gibbon? Okay, I heard can't hear. Let's see. Hear that time? So it is a gibbon, but it's a male gibbon. And then, oh, can't hear. Let's see. Interesting. Seemed like some of the other ones were working for some reason. Can you hear this one? Okay, okay. So since that wasn't working, can't hear it, can anybody guess based on the spectrogram? You've seen a lot of the female gibbon spectrograms. What do you think? Yes, no? Yes, it is. All right, that's interesting that some of the sounds were working, but some weren't. Okay. And so as I mentioned earlier on in the talk, we had the idea between supervised and unsupervised learning. And so I showed you how we have used supervised learning to distinguish between different individuals. And now I'll tell you kind of how we have used unsupervised learning to distinguish between individuals as opposed to supervised learning. And So I've mentioned this a little bit already, that as a field in bioacoustics, we are really kind of starting to see a shift between from using these like handheld battery operated recording devices to setting out these autonomous battery operated recording devices that can go for really, really long periods of time. And so this field or this approach is called passive acoustic monitoring. And so we have, our lab has a few projects going already. So we have one that's led by Dr. Wendy Erb that's in Central Kalimantan that's been going for quite a few years where she's been collecting continuous data. And then I have some recorders out, or they're not out now, but they were out in Danam Valley, and we're hoping to get quite a few more out across peninsular Malaysia. And so this is an area where I think that hopefully out of this seminar and the networking here that we might be able to get some passive acoustic monitoring going in peninsular Malaysia. And so with passive acoustic monitoring, there are a whole suite of benefits to this. So first of all, we can collect continuous data in the absence of human observers. So we can collect data at night. We can do all of these things where, you know, we don't need a human there. And you can basically go and set them, forget about them, and then come back. But there are issues, particularly when you get that data back of, okay, what do we do with all of that data? And so I already talked about this bullet point. So first of all, what do we do with all of this data? So in the case of the gibbons, thinking about if we have continuous data going for 24 hours a day for months at a time, we either need to have human observers go through and look at the spectrograms and pull out the calls, or we can do some combination of automated detection, like the detector I just showed, and then have humans go in and verify a little bit. But the main difference here in this data set compared to the supervised data set that I showed you before is that because there are no humans there, we don't know who is calling. And so in this case, we lack what we call labeled training data regarding caller ID. And so as I mentioned before, gibbons are territorial. And so that does help us a little bit. So we know if we place one recorder in one place and then another one 5 kilometers away, that the gibbons that we record on those two recorders, because of the home range size of the gibbons, they're going to be different. But there are cases where you may have a recorder that is set at a territory boundary, and where you may have multiple individuals from two groups calling, but you don't know who is who because there's not an observer there. And so there are ways that we can get around some of these issues. So first is acoustic localization. And so in this case, we need to be able to detect the sound, in this case, the calling gibbon, on at least 3 of our recorders. And then we can use a approach called triangulation. And so based on the difference in the arrival of the sound of the calling gibbon to the 3 recorders, we can localize where the animal is. This is really something that has been developed really well and works quite well in the marine environment. And so as I mentioned before, sound travels much, much easier, much further in the marine environment. I tested an acoustic localization system that we developed in our lab out at Danam, and it was really challenging. There was a lot of work that went into it. It was really, really hard. And the main reason behind that is there's something in the autonomous recording units, it's called clock drift. And so in this case, we set our recording unit to have a certain sampling rate. So, collect 44,000 samples every second. But as the time kind of goes on, the clock may shift just a little tiny bit, which doesn't matter for a lot of our applications when we're thinking about, like trying to find gibbon calls in there. But if we're using the acoustic localization, we have to have really, really precise down to the millisecond differences in the time of the arrival of the signals. And so if we have this clock drift that's starting to happen over time of these different recorders, that's also going to make acoustic localization really, really tough. If you can you can avoid this by using GPS synced units, but those are quite expensive. And then again, the processing of the data afterwards is quite hard. So anyways, that was a very long way to say that acoustic localization exists. It's quite well used in the marine systems, but in the terrestrial systems, it's really hard. It's much harder. But so coming back to this idea of needing to have an idea of the number of individuals, a lot of these conservation applications, in particular density estimation, requires us to know how many animals are calling. And so with passive acoustic monitoring data, it can be challenging if the observer is not there. And so in come unsupervised methods. And so that's really kind of the whole justification for why we are interested in using unsupervised methods in passive acoustic monitoring data. And so my postdoc advisor, Holger Klink, and myself, we got a paper out last year in the journal Methods in Ecology and Evolution, where we tested 3 different unsupervised clustering algorithms, and we compared the results of those unsupervised clustering algorithms to the results of our actual data set, which this may be a little bit counterintuitive. So we used, as I mentioned, 3 different unsupervised algorithms. And so from my dissertation, we had a labeled data set of 883 calls from 53 gibbon females. And so this may seem a little bit, as I said, counterintuitive, the fact that we wanted to have a labeled data set for unsupervised methods, but actually it's really, really useful to validate the output of the unsupervised clustering algorithm. So that's why we use this data set. And so with this expanded data set, and so this was 53 females from different sites across Sabah, so not just from one site like the data that I showed you earlier. And so using supervised methods, we could get a classification accuracy of greater than 90%, which for this really high sample size, and if any of you are a primatologist or primate behavioral ecologist, you know that our sample size is generally like 1 or 2 animals. So to have 53 females, that's a really fantastic, big, large data set. And to be able to classify to the individual with a 90% accuracy, that's showing that there is a really huge degree of individual variation there. And so here we have some UMAP plots. And so UMAP is it's a shortened thing that you don't need to worry about what it stands for, but it's a data visualization technique, and it's kind of one of the more common ones that's being used now. And so it's the same kind of idea as I was talking about before, where we have for each call, we have a set of features, and then we input that set of features into the algorithm. And so in this case, if you look at the plot A, we have each point represents 1 call from 1 gibbon female individual. And then the different colors are showing different gibbon female individuals. And so again, since we see this really great separation of the females that are coming out from different females, this is showing basically just a nice data visualization technique. And so that's the plot A, so the one on the top left. And then plots B through D, we did this as a way to show whether the unsupervised clustering algorithm assigned a particular call to the cluster that we matched with the female ID. And so in this case, if you look at affinity propagation clustering, it did a really pretty good job. So it assigned with an accuracy of about 65% correct. And so again, this is not giving it any training data. This is basically putting in your data, saying find the different clusters, and tell me how many clusters are there, and then assign each point or each call to a cluster. And so we found for affinity propagation clustering that it was about 65% correct. Gaussian and mixture models were 53%, so decent. And then K-medioids, you may have heard of K-means clustering before. So K-medioids is similar, it's just a little bit different in the underlying math. And so it got 17% correct. So was not doing so hot. And so we did a set of random iterations where we randomly chose calls from either 10, 20, 30, 40, or 50 females. So that's what we're seeing on the x-axis. And then we calculated our percent correct classification. And so basically what you're seeing on the top plot is kind of similar to what I was showing you in the last slide, but we ran it over multiple iterations and had it assigned to each call to the cluster, which we matched to female. And so the big takeaway from that is that affinity propagation clustering, which is shown in yellow here, across the different numbers of females, it was really quite consistent in how well it was classifying individuals. And then if you look at the K-medioids that goes down, if you look at when we had 50 females, the K-medioids was not doing very well at all. It was not classifying many females to the correct cluster. But when we're thinking about the applications for passive acoustic monitoring, like what we really want to know is not how well can you match each call to the respective female, but what we care about is in this data set, if there are 20 females here, how well is the algorithm going to tell me about that there are 20 females there? And so that's what this the bottom plot is telling us. So we were interested to know the deviation from the actual number of females. And so all three of these unsupervised clustering algorithms, they run and then they tell you how many clusters there are in your data set. And so then we would say, okay, so for the first one, we put 10 females. We just randomly chose 10 females and all of the calls from 10 females. And then we put it into the algorithm. We say, how many clusters are there? And so a deviation of 0 is actually what we want. That's really, really good because if it's telling us that there are 10 females when there are 10 females. And so then if you look across as we move up from the number of females that we include in our data set, again, affinity propagation clustering did really, really well. And so that really, like for the passive acoustic monitoring application part, the affinity propagation was the best. And it was the best in terms of returning the number of females or the number of clusters that best matched the number of females. And so I think just because of, time constraints, I will shift through to conclusions. And so in conclusion, we have that I hope I have been able to convince you that both supervised and unsupervised approaches are important for studying primate vocal behavior. And again, the supervised and unsupervised approaches are not just something that we use in primatology. Like these are machine learning kind of across the board, but there are some really interesting applied questions that we can ask. And then the availability of the labeled training data is going to be the main factor when choosing among our different types of approaches. And so as I showed you for my dissertation, I was out there with a handheld recording device. And so I was able to know which female was calling at which time. So I have that labeled training data set. But now that we're shifting towards the use of passive acoustic monitoring, where we don't have human observers there, we're not going to have labeled training data. So there is a really big shift in people being very interested and wanting to know if we can use these unsupervised approaches to give us information about the number of individuals or other things that are existing in our data set. And then lastly, so I showed you the results of the paper that my postdoc advisor and I did. And as I mentioned, we had labeled training data that we used to kind of validate the unsupervised clustering results. And so now the next steps that we need to do is we need to kind of go out into the real world and have human observers there, like with a big grid of autonomous recording units, and kind of do the whole data analysis pipeline. So record for 6 months, pull all of those recorders in, do the automated detector, have it tell you how many individuals are in the area, and then we actually need to have the humans go and validate that. So we're still at this point where we're developing the methods. We still need human kind of observer and input, and then before we can actually put those into practice, there's still more work to be done. So. And with that, I will go ahead and finish up. And thank you so much for your for listening. And if you had any questions or anything that you'd like to talk about, please feel free to send me an email here. And I did see that some questions were coming up in the chat as well, so I think that we should have some time for those. And again, I apologize for the technical difficulties, and thank you for being so patient about that too.
All right. Okay, thank you, Dina, for a really eye-opener. How application of machine learning can be significant contribution to primatology and anthropology. Okay, so we go straight for the question and answer session. We have, okay, let me just read. We have from Dr. Suryati Jamil. Hi, Dina. My name is Riza from Computer Science Department. We are just starting our research in acoustic classification of lowland forest bird. Okay, there's a few questions. Maybe we can one by one, right?
Yeah, great.
Okay.
Yeah.
Can you inform us of any challenge you face during data collection? How best to place the acoustic device to reduce noise and physical disturbance? That's the first one.
Okay, so, and this seems like it's a question related to data collection to start. And it's a lot of it comes down to basically doing field tests. And it also depends where in the canopy your target species are. So if you're interested in species up in the canopy, then you probably want to put your recorder up in the canopy. Whereas if you're interested in species that are kind of closer to the ground, then you might want to do that. My experience with gibbons, and again, like their calls, you know, there's the gibbons, the Argus, and the hornbills. Like their calls, they're long distance calls, and they travel for kilometers, you know, over the scale of kilometers. And so the placement of the recording unit within the canopy is a little bit less important, just because you're still going to be detecting those animals. But with a lot of birds, because they're small, but then also the sounds that they're making, they're not necessarily meant to like travel for kilometers. And so you probably want to get your recording device up kind of closer to where they're going to be. And that's something that will be basically kind of, you'll need to field test that. I have been really lucky. Like I've had recording units in Danum Valley that like none of them got bothered by anything, and they were just at shoulder height. We had elephants that were around for quite a bit that I was a little bit concerned, because I know that with camera traps, the elephants can sometimes come in and destroy them. And so, I mean, I guess it also depends if you're worried about animal disturbance or if you're working in an area where there are a lot of people around. In that case, if you're worried about that, I would suggest probably trying to get them higher up in the trees. But and it sounded like there was more.
Okay, there's okay. The follow-up question is where is it? Oh, there's some question coming drops. Okay, by other animals such as squirrel and monkeys. The follow-up question related to that. How do the data collection to place the acoustic device? Do you have experience with that?
For squirrels and monkeys, you know, not targeted. In Danum, we definitely got a lot of the leaf monkeys because they have that funny like kind of sound. Squirrels, I'm sure that we have them. I just haven't looked to see. And so, I mean, I guess, you know, it depends a lot too on kind of the questions that you're interested in as well. So, you know, if you're interested in trying to figure out kind of all of the species that are in a particular area versus if you're trying to target one species or another, that's going to influence not only like the placement within the canopy, But then another big question when developing a passive acoustic monitoring project is how you space the recorders across the area of interest. And so if you have them at 500-meter spacing, a kilometer spacing, or 5 kilometers, and so it's really, that's going to depend a lot on your target animals and then also your research questions. And so I'd be happy to chat more about that, like if the person wants to send me an email.
All right. Okay, good. Another question: did you?
Okay. So some of you probably
someone can mute that.
Okay. So these are examples
Dr.
of
Valayan.
Okay.
Hello. Okay. Okay. Another question: did you do any pre-processing prior to the feature extraction?
Yes. And so that's a really great question too. And so for the project that I showed you with my dissertation, I was out and I was using the handheld recording units, but they were only going for 20 or 30 minutes at a time. So I didn't have to browse through hours and hours of data. But you do have, because the gibbons, as I mentioned, they have these duets. So the males and females will go back and forth. And so I had to go through and visually scan the spectrograms. And then basically using this program Raven, you can draw a box around all of the spectrograms or signals of interest. And then you can use Raven, or I use R, and you can pull out all of the sound files that are just the gibbons or just the signal of interest, and then do all the other processing in R or any program that you want. And so, yes, there is generally a lot of pre-processing with these things. And spectrograms, it's interesting because it gets to be like reading music, where like I can scan through hours and be like, not a gibbon, oh, that's a gibbon, like it becomes second nature, but it does take time to get used to reading them.
All right. Okay, good. Okay, we have another question from Dr. Wahida. Hello, Dina. Thank you for your presentation. My research lab is into sound absorption of typical plant in Malaysia. I have collected data using in-house built measurement tools. I'm not using spectrogram. Any affordable option other than spectrogram or monitor for the noise, for sound? Anyone from your lab working on the same thing? That's a question.
So if I understand correctly, it's sound absorption of the vegetation. Is that right? Of plants? Yeah.
Yes.
That's amazing. That's something that we are all very animal focused in our lab, but we are currently working on a project where we basically playback experiments. So we broadcast signals using like a speaker, and we re-recorded them at different distances, and we did that across different sites. And so it's a little bit different, I think, than what they're doing, but it's also a really big question that we need to answer if we're going to use passive acoustic monitoring is, how far are these sounds traveling? How far can we detect them with these recording units? But then coming back to the question of the sound absorption by the plants in a lab setting, I guess I would have to know kind of the design of the study and the structure of the acoustic data. So I imagined you would maybe be broadcasting a sound, and then you have the plant kind of in between the speaker, and you're either re-recording using an acoustic recorder or a sound level meter. Or something along those lines. And so it would really depend kind of how you were collecting the data, who would subsequently analyze those. But that's something that I think is really interesting, and not anything that in our research lab that we've been able to get at, but it's really crucial to know, like across different forest types, across different like vegetation types, how is sound traveling? How is it being changed by these different factors? So I think that's a really interesting and important thing to be looking at.
All right. Okay, thank you. Okay, the next one is, does gibbon vocalization also a way to mark their territory?
Yes. Oh, that's a fantastic question. So because gibbons are territorial, we do think that these loud calls, that they serve at least partly a function of saying to other gibbon groups, like, this is our spot, stay out of here, like we're happy here, stay away. But there are other hypotheses as to why we see duetting. And so to just give a little bit of background, so in non-human primates, we see duetting kind of across the orders. So we see in Indries in Madagascar, in tarsiers, which are found in Southeast Asia, but particularly the ones on Sulawesi. We see in titi monkeys, which are found in South America, and then also in gibbons. And so it's not really common because there's hundreds of species of primates, but it's like throughout the primate family tree, if you will. So the other kind of interesting thing is that in all the primates that duet, we see they're pair living. So it's just the male and the female with their offspring, and they're territorial and they duet. So we think, there's something that's happening where we see this interplay between the social organization and them being territorial and them engaging in these duets and these loud calls. But then coming back to the idea of the functions, we think, and we're not sure, like people, and this is something I'm hoping to be able to do hopefully sometime soon, are doing what are called playback experiments, where you go into the presumed territories of gibbons and you play either a male solo or a duet. You can play a neighbor or a gibbon they've never heard before, and you can see if they're responding differently to try to test some of the hypotheses. And so another one that stands out to me as there is some evidence to support this, it's called the pair bonding hypothesis. And so the idea behind this is that duetting is similar to grooming or other types of behaviors where the male and the female, they're kind of reinforcing their social relationship. And so in duetting, that's also, we could think of it as being analogous to grooming. And so there are, these kind of different hypotheses as to why they're doing it. And it's for these really complex vocalizations to have evolved, like they have to have some sort of function, it's like it's some sort of really important function. So that's a really great question. So thank you for asking.
All right, good answer. Next question from Dr. Rizauddin. Is it possible if you could demo the Gibbon R package that you have developed?
Oh, yeah. So, so the Gibbon R package is really the automated detector that I showed you. It's kind of the foundation for that package. I don't have a setup right now to show for the Gibbon R, but I saw that question in the chat. So whoever asked that, they can feel free to send me an email, and I'd be happy to talk them through that, because it's still very much in the development stages. One of my goals for the next year is to actually try to get it out published somewhere. But it's very writing our code and then getting it into a package that other people can use. They're two different things. So I'm still learning how to navigate getting all of the bits working for the package and everything. But
all right. So next from Dr. Hyo Chong Chin. Hi Dina, great presentation. My name is Chin from the Faculty of Medicine. I'm an entomologist, graduated from Texas in 2016. My question is, can you use acoustic signal in studying insect biodiversity?
Oh, that is a great question. Yes. So we have, Dr. Laurel Symes is the assistant director at our lab, and she works a lot with katydids. So they're a type of insect, and they're found, I know she's working in Central America. I think they're found kind of all over. And so they are doing, it's a really cool project. So she's working in collaboration with some of the engineers at our lab to develop basically kind of classification or classifiers for, I want to say that they're up to 100 different species of katydids right now. And so, yes, the insect acoustic diversity is something that our lab is really, really interested in. And so I would be happy to put you in contact with Dr. Symes. She's really wonderful, really approachable, and doing really, really fantastic work. And we've been thinking about how we can try to expand to do some of the insect work in Southeast Asia, but obviously that's very far from my area of expertise, but it would be just fantastic to try to include some acoustic stuff with that. So yes, thank you for that question.
Okay, we have one more follow-up question from Dr. Saleh Daim. He asked, can he ask it live? Who's the admin? Can you unmute Dr. Saleh Daim? He wants to ask it live.
Yeah, hello.
Okay.
Hi.
Hello. Hi, Dina.
Hi, selamat pagi.
Yeah, selamat pagi. Very interesting approach in the new behavioral study using acoustic. I'm actually interested. I'm actually in biodiversity management. I'm not an expert in certain species, but I'm more into biodiversity management. And currently, I'm looking at habitat suitability, and I have a PhD student who's now studying habitat suitability modeling on tigers in Peninsular Malaysia. And I have followed the gibbon in the peninsula for quite a while, and I've been attempting to do some behavioral study, but no graduate student takers. You know, it's so difficult, and they don't want to jeopardize their their studies. No, it's not easy to study gibbon. I know, I understand that. But my question is, in terms of biodiversity management, in terms of habitat suitability, I understand that you use acoustic, you use vocalization, and it's a territorial marking, and you use triangulation. My question would be, as for now, your study in given in Sabah with all the method you use, can you have you able to sort of estimate the size of a territory from a single group? Because it is very important to, when we are looking at biological corridors, buffer zone and all that, and using gibbon as indicator. And in Peninsular Malaysia, study on gibbon is very limited. I think the last published is in 1957, I think, on gibbon. And there's a lot of gibbon studies in Indochina up to until Thailand, but not in the peninsula. And interesting to know that there are studies like yours in Sabah. Very interesting. Congratulations. So can you explain that? Are there any estimated territory that we can use from this vocalization so that we can assume the suitable habitat for gibbon? Thank you.
Yeah. So that is an approach that we are very interested in working to develop. And so I know in Africa, they have used autonomous recorders to track chimpanzee ranging. And so I know chimpanzees, obviously, they range over a much larger area. So that is something that people are already starting to think about. With the mapping of the territories, it's definitely feasible and possible. And we are actually about to start to pilot work doing just that in Cambodia. But the reason that we're going to be doing it in Cambodia is because they have habituated gibbon groups. And so we basically need to be able to validate the acoustic data methods with actual methods where people, so the traditional way that people estimate home range size is to do full day or, like behavioral follows of the gibbons as long as they can. And so that's something that we're going to be testing out and trying to see basically what you just asked, like, can we use a grid of, and we would have to put the recorders really close together. So can we use a grid of autonomous recorders that are quite close together to basically map out the gibbon territories or home ranges? And I mean, it would have to be based on their calling only. So assuming if they're calling at the territory boundaries, but it's feasible. It's still in the development stages, and it would require some validation and other types of data collection. And particularly, even if we pilot in Cambodia, the forest is going to be different. They may actually have different home range territory sizes as well. So I think if you were interested in trying to do it in Peninsular Malaysia, we'd have to kind of start with that pilot work and then potentially move out to have a much broader kind of grid of autonomous recording units.
All right. Okay. Thank you. We have next from Dr. Velayan.
Yeah. Can mute me?
Yes, you already unmute. You can ask Dr. Dina now.
Okay. I have got a few things to contribute to you. I'm a bit sorry because I am on 3 webinars, one on coronavirus in animals, one on the Fulbright, which is in my heart, and the other one is the brain workshop that's going on in UiTM International for the last 4 days. I'd like to put certain things in order. I do not know whether you know the history of the study of gibbons or primates in this country. Just now, Saleh was saying that Saleh Dine, that the informations were way back in 1957. But we had a project under the NIH and the University of Cambridge to study the gibbons in detail, both the siamang and the la gibbon. In fact, I was one of the person who did my master's in the gibbon nutrition at Kolakrai, the work which was done by late Raymaker in 1977. And I went and re-identified the trees. I did a lot of laboratory work and field work with the gibbons. And I know David Kivers has worked extensively on the gibbons in Peninsular Malaysia. They've never explored. I don't know why these data are not being remembered, or the population of the primates were also census were done by late Marshall, So there were 3 other students from the UK benefited from this study. And we had a large center of studying the primates in UPM at the time, and then the center could not proceed because of lack of researchers, etc. My question is, are you aware of all this which has taken place in Peninsular Malaysia? Question number 2, you are saying you have studied the gibbons in Sabah. In Sabah, I'm sorry, I couldn't hear any of the vocalization and your your lecture was very distinct to me. Is it the Mulleri gibbon or what gibbon it is? Please specify, because there are other species of gibbons in Malaysia, and which region and which species you really worked on it. Thank you for answering.
Yeah, well, so at the start I mentioned, so it's the northern gray gibbon. So in Sabah, that was just recently made into its own species. So they all used to be Hylobates mulleri, but now they've been separated out into their own separate species. So it's in the northern part of Sabah. And yes, I mean, I definitely, in my, dissertation and in papers that I write, I always try to cite, the literature, the early work on gibbons. And even, like John Matani in the 80s was working on gibbons in Sabah and Borneo. And then, and I'm familiar with some of the other ones, Raymaker, I believe you said. So yes, we do try to look and see what's been done in the past for sure. So. But a lot of the work that I'm doing now, a lot of it is using kind of newer technologies, and so we use the older papers to cite for kind of the important behavioral information and natural history and things like that. And now then the stuff that I'm doing, it's only in the last few years because of the technology and the analytical tools that we have. So.
All right. Okay. Okay, I think we have to stop here. We are past our scheduled time. If any queries, you can email Dr. Dina straight. I think Dr. Dina can type her email in the chat box if it's possible. If someone else want to email you related to this question. All right. Okay, thank you very much all. On behalf of the organizer, the Office of Deputy Vice-Chancellor Research and Innovation, UiTM Research Nexus, and FAAM, I would like to thank our guest speaker, Dr. Dina Jane Klink, and all the audience here for making this wonderful program a success. So for now, thank you so much for your time, and we hope to see you in future event. Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much.
Dina, I think you can check in the link.